The first speaker is Shannon Boye from the University of Florida, who will be talking about AAV vectors in the eye.
Thanks, David. Can everyone hear me okay? Okay. Well, good morning. I'd like to thank the organizers for having me here at 8 a.m. on Saturday morning. I'm happy to see so many of you here as well. So I'll be talking to you today broadly about AAV vectors in the eye, and we'll keep it sort of basic and educational, and I'm happy to answer any deeper, more specific questions at the end. These are my disclosures. Okay, so if you have been at this meeting for the past few days, few years, you definitely know what AAV is. But this has become the gold standard in retinal gene therapy and ocular gene therapy and even outside of the eye due to many of the features that you see listed here, including that it's non-pathogenic, relatively non-immunogenic. It can deliver its payload where it remains in episomal form inside the cell, and the transgene expression will persist over the lifetime of that cell. What's great for us in the retina is that AAV has the ability to transduce non-dividing cells very effectively, which is of course important for postmitotic neurons. And it also comes in a variety of flavors or serotypes. Most importantly, however, is that it's clinically relevant. So I've listed for you here all of the diseases for which AAV has been used in a clinical trial. These are either ongoing or completed. And of course, everyone in the room knows that the RP65 form of LCA2 just had a gene therapy approved for its treatment just a couple of months ago. So it's a really exciting time in the field of retinal gene therapy. So for the non-ocular folks in the room, just very briefly, up on the top left, you have a cross-section of the human eyeball. Light comes through the clear cornea, and it's focused on the retina with a structure called the lens. And despite its small size, the retina is actually pretty complex. There are several neuronal cell layers, and light has to travel through all of those layers before it falls on the photoreceptors. And the photoreceptors are really ground zero for vision. Yeah, you're right, this doesn't work at all. Okay. So we have two major types of photoreceptors in our eye, rods and cones, and they're distributed in a very specific fashion in the vertebrate retina. So our rods are concentrated in the peripheral retina, whereas our cones are concentrated in the central retina in a region that we refer to as the macula, and at the very center of the macula is the fovea. And as these cartoons on the right depict, what we really use our rods for is for vision under dim light circumstances, whereas cones are very important for you to be able to see the screen right now, to see color, for high acuity vision. So not surprisingly, based on how many proteins are expressed in photoreceptors that are important for our ability to convert a photon of light into an electrochemical signal that's eventually sent to the brain and processed as vision, the vast majority of inherited retinal diseases are caused by defects in those outer retinal photoreceptor cells, including some of the diseases I've listed to the right. So the current state of the art for getting these therapeutic genes into those photoreceptors is a subretinal injection, wherein a needle is placed between the neural retina and the retinal pigment epithelium and deposited right next to those really important photoreceptors. This is a fairly invasive procedure. It does pose some risks, but it has proven well tolerated in several trials. But it does involve a complicated surgery, a dedicated surgical team, a special facility, so it is not a simple procedure. So there was one red flag that came up from one of the groups that performed an LCA2 phase 1 clinical trial. And so this was from the University of Florida, University of Pennsylvania group. And what Dr. Jacobson noted was that when certain patients received subretinal injections under their cone-rich foveas, there was actually a loss of central retinal thickness, and in some patients a loss of visual acuity. This wasn't seen across all of the trials, but it was seen in this. And so it raised a red flag for the retinal gene therapists in the field that perhaps we needed to think a little bit more, thank you very much, to think very carefully about the consequences of detaching that cone-rich fovea. And so that led my group, as well as many other groups, on the pursuit to develop a less invasive surgical procedure. So instead of placing the vector between the neural retina and the RPE, which involves a retinal detachment, what if we could instead place the vector into the vitreous, which is sort of the liquid-filled globe part of the eye, and have those vectors penetrate back to those all-important photoreceptor cells. And our belief and our hope was that we could convert this relatively detailed procedure into something that could be done in an outpatient setting. So what I'm showing you here, you obviously don't have to memorize all of the details, but this is circa 10 years ago. So we had these AAV flavors are serotypes at our disposal, and they had all been well characterized for their transduction profiles across multiple species. And you can see that there's much more present on the subretinal table, and that's because not much happens when you put a lot of different AAV serotypes into the vitreous. But the point of this slide is just to tell you that this was just the beginning. As you've seen in this conference this year, as well as years before it, there are new AAV capsid variants being developed and designed every day at this point. And the reason for that has to do with the AAV capsid being so malleable. So you can see here, this is an image from my colleague Mavis McKenna's lab. The AAV capsid contains loops, which are these little bumpy things you see on the outside of the capsid. And we know that there are 9 common variable regions in AAV, and that the sequences in these variable regions dictate the functional differences between AAVs. So why is one AAV good at getting into photoreceptors and another is not, for instance? So the question we ask is, can we modify the sequence of these variable regions in order to generate new AAVs with desired transduction profiles? So in our case, we want to design AAVs that can get to the photoreceptors following intravitreal injection. So there's a couple strategies that have been used to do this by multiple groups. The first of which is rational design, and this is a technique that was pioneered by my colleague at UF, Arun Srivastava. And basically what he did was he used his knowledge of vector biology to make structure-informed mutations to that capsid. And so an example of this is that Arun knew that when an AAV vector gets inside the cell, there are residues that become phosphorylated, and that signals the capsid to be ubiquitinated and ultimately degraded in the proteasome. And so first he used drugs to prevent that phosphorylation and showed that he could reduce degradation and increase transduction. And then he reasoned, why not mutate those tyrosine residues to begin with so that they're never phosphorylated? And so this formed the basis for the now widely used rationally designed YF capsid variants. So again, this is an example of using your knowledge of vector biology to make structure-informed changes to the capsid. The other method that's been heavily employed, and you've seen many examples of it over the week, is directed evolution. And put very simply, what this means is that you take a whole bunch of stuff, you throw it against a wall, and you see what sticks. That's a very simplified definition. But it doesn't require any mechanistic understanding of the barriers that are associated with getting to the photoreceptors from the vitreous. You're simply asking nature to tell you what is the best sequence, what sequence confers that capsid with the best ability to get to those photoreceptors. So I'm showing for you here the result of many years of work from multiple groups, but this is an example of a rationally designed capsid variant, as well as one that was mined from directed evolution of an AAV2-based capsid library. Now, both of these variants have shown quite a bit of promise following intravitreal injection in mouse retina. So you're seeing pan-retinal, outer retinal transduction following intravitreal injection of either of those two capsids when they're injected into a mouse. The problem has become that when you put those very same capsid variants into a clinically relevant species, into non-human primate, and remember, non-human primates and mice have very different eyes. The hurdles that exist in non-human primate eyes are quite formidable compared to what is present in mouse eyes. And you can see that the transduction profile of those vectors is quite different when they're put into a monkey compared to when they're put into a mouse. You have this expression that's restricted to this ring of cells around the fovea, some cells in the foveal pit, and then some cells sort of in punctate regions in the periphery around what we think are blood vessels. So the signal to everyone that there was still work to be done. And so something that we thought in my lab, and other labs have thought of this as well, to be fair, is that many of the variants that you've seen that were originally identified and performed well in mice were actually mined using directed evolution in mice. And mice, again, don't have the same barriers to transduction that monkeys do. So we thought it would be worth our time to screen these capsid libraries in the monkey itself. The problem being, of course, that we don't have monkeys walking around with glowing green photoreceptors. If we did, that would be beautiful. If anyone wants to pay for it, let me know. But we had a tool with which we could do that. So AAV5 GRK1 GFP, we've shown in the past, can specifically label rods and cones following subretinal injection in non-human primate. And so what we did in collaboration with Paul Gamlin at UAB was to subretinally inject multiple blebs of AAV5 GRK1 GFP to label as many photoreceptors as we could. And then Paul was also familiar with the technique wherein you could inject microRuby into the lateral geniculate nucleus, and that would retrogradely label the ganglion cells. So now we have monkeys with sortable photoreceptors and sortable retinal ganglion cells, into which we could ultimately inject our capsid library. We had to do some quality control first, so this is just an example showing you that we can create multiple subretinal injection blebs in a monkey with that AAV5 vector. And here's an image showing you a cross-section through the brain and showing you that the LGN did receive that microRuby. This is an example showing you that indeed the retinal ganglion cells were retrogradely labeled by that dye in the LGN. And then what we did was we needed to confirm the purity of those sortable cell populations. So we took cells from the central, the macular retina, the superior and the inferior retina, and we performed transcript analysis. Of course, we sorted out the GFP-positive photoreceptors and the microRuby-positive retinal ganglion cells. This is a very busy slide, but I'll just take you down to one example here. So in this example, we're looking at the GFP-positive cells from the central retina. And you'll note that they're enriched only for photoreceptor-specific transcripts like cone transducin alpha and rhodopsin, whereas they're de-enriched for things like GRIM6, which is a bipolar cell marker. So the quick summary of this slide is that we were able to validate the purity of our cell populations using transcript analysis. So at this point, it was time to throw in the capsid library. And to do this, we relied on Sergei Zolotukhin, another wonderful institutional collaborator at UF, who had at the time designed a highly complex AAV2-based library, complexity about 10 to the 8th, and it was a structure-informed library in the sense that he only made changes in regions that he knew wouldn't conflict with changes in other regions, such that all of the capsids that were produced were actually packageable. Okay, so what we did then was inject the capsid library into the monkeys that we had made sortable. We performed a first round and then separated them out into two sub-libraries, one that was photoreceptor specific and one that was RGC specific. Performed additional rounds of screening and then bioinformatic analysis out the back end and also characterization of the enriched variants in mouse to begin with. So I'm going to jump to the chase and just show you a few of the variants that came out of this screen. So what we did was we intravitreally injected a mouse model with glowing green photoreceptors with 2e⁹ total vector genomes of self-complementary AAV, small CBA, M. cherry. And you can see that relative to the benchmark vectors, the quad YF T2V and 7M8, that we do have variants that are highly transducing the mouse retina following intravitreal injection. The next question we asked was, well, can we further improve this with rational design? So even though these libraries are very complex, there are still not an infinite number of possibilities contained within them. So we thought that we could add to this by incorporating some of those YF changes that I spoke about earlier. And the answer to that question was yes. You can see that one of the variants mined via the library screen that had YF and threonine to valine mutations added exhibited further improvements in transduction. And this is just a pictorial summary of all of the variants that we've looked at so far relative to the benchmark vectors on the far left and the far right. And again, I'm not showing this data, but we could quantify these results because they were done in the NRL GFP mice, which have sortable green photoreceptors, and there were significant improvements in the transduction of these variants relative to benchmark. This is just an example showing you what things look like in cross-section. So when you use a ubiquitous promoter and you inject one of these strong variants, you can see that basically every cell type in the retina is transduced. I've blown up the contrast here so you can appreciate what's going on in the outer retina. When you pair that variant with a GRK1 or a photoreceptor-specific promoter, you can see how robust the photoreceptor transduction is. Okay, so the next step obviously is to inject these enriched variants in monkeys. I'm an academic lab, not a company, so we had to find sort of a cheaper way to do this initially. So what we did was combine all of the enriched variants, each of them containing a unique barcode that we could look at after we sacrificed the animal, took the tissue and did deep sequencing. And that would allow us to tell which of these variants was most enriched in that target tissue. So I'm just going to show one example of one of the subjects. Up top you can see the AAV5 mediated GFP expression from making the primate sortable. And on the bottom you can see that very classic transduction in the ganglion cell ring, the foveal pit, and in these peripheral areas. And the point I want to make here is that obviously this is a drastic improvement over what's achieved with just plain unmodified AAV2. But there are still things missing, and I think that this is similar to what other groups are showing. We are still not achieving the pan-outer retinal transduction with these intravitreally delivered vectors. So my personal belief is that yes, this might be good for targeting central cones. I think we're going to have to focus a lot of attention on the dose that's used and any potential inflammatory responses, but I still think that there is work to be done in this field in terms of generating a virus that can be used to target the outer retina across the retina from the vitreous. So we wanted to understand why. Why are the vectors that get back to the photoreceptors from the vitreous doing a better job of that? And because if we can understand the mechanism of this, then perhaps we can harness it and make these vectors even better. So to do that, we needed to understand the barriers to transduction via the vitreous, the first of which is a dilution effect. That dilution effect isn't as big in a mouse, obviously, but when you put this vector into a monkey eye, it's pretty big and there's a lot of vitreous. Another major barrier is the interlimiting membrane, which is a typical basement membrane formed by the end feet of these Mueller glial cells that traverse the entire thickness of the retina. And so when you put the AAV into the eye, it slams into this interlimiting membrane and it poses both a physical and a biological barrier, which I'll discuss in a moment. So what do we know about the interlimiting membrane? Well, we know that AAV serotypes, I should say this is work based on Denise Del Cara, where she took AAV capsids and she fluorescently labeled them, and then she injected them into the vitreous and looked to see which capsid stuck to the interlimiting membrane. She found that AAV2 and AAV6 stuck very effectively. AAV5 and AAV8 not so much, AAV8 just a little bit. But what she hypothesized was that this was receptor-based. So AAV2 binds heparan sulfate proteoglycan, which is very abundant in the interlimiting membrane. AAV8 can bind laminin, which is also present in the ILM, not to the degree that HSPG is. But AAV5 binds sialic acid, which is absent from the ILM, so that you can see that there's a receptor biology component here. The capsid needs to be able to stick to that membrane if it's going to move past it and get into the retina. So together with Mavis McKenna, we evaluated some of our variants. So we had a series of variants that we knew were okay at getting into the retina and some that were very good at getting to the outer retina. And we probed their heparin binding affinity. And what we found was that the variants that were best at getting to the outer retina via the vitreous had slightly reduced heparin binding relative to their unmodified parent serotypes. So they definitely bound heparin. I don't want to convey the message that they don't need to bind heparin, but they bound it a little bit less tightly. So then we looked at those variants, and in this experiment we included a variant that is completely deheparinated. So its canonical heparin binding residues have been mutated, and we tested them following either intravitreal or subretinal injection in mouse. And we confirmed our hypothesis that heparin binding was required for transduction of the retina via the vitreous, because that deheparinated mutant does nothing when you put it in the vitreous. However, much to our surprise, we found that when we put it subretinally, it was gangbusters, and it also had this increased lateral spread relative to the other variants that we had been testing. And subsequent to this, we've had colleagues test this variant in the brain. It also exhibits increased lateral spread there. And what we did was we quantified the transduction of this particular variant relative to AAV5, which is a very typical serotype used to target photoreceptors subretinally, and we found that it was actually more potent than AAV5. So what this tells us is that heparin binding is important for these variants to get to the outer retina from the vitreous. You need to be able to bind heparin, but the ones that get back most effectively bind it a little bit less effectively. It's sort of a seesaw phenomenon. You want to bind, but you don't want to bind so tightly that you get stuck at that basement membrane. But heparin binding is not a requirement for photoreceptor transduction, and we proved that by subretinally injecting that deheparinated variant in the mouse retina. So we thought to ourselves, we're pursuing finding an intravitreal vector, but it's always good to try multiple things in case one doesn't work. So knowing how much of a hurdle the interlimiting membrane is to transduction via the vitreous, my colleague Paul Gamlin and Doug Witherspoon at UAB and I decided that we would try and bypass that physical barrier altogether. And we did that with a novel injection technique called sub-ILM injection. So in this procedure, a needle is placed between the ganglion cell layer and the interlimiting membrane. And what we do is we put a little bit of viscoelastic in that space first, and then come in with AAV, and a bleb can be created in that space. And what we showed, this is a preliminary study, it's published, but we first just used AAV2, plain AAV2. And we showed that the area of transduction following a sub-ILM injection was much better than what we would get with an intravitreal injection, which I've shown you down here for reference. And you can see, when you look at all of these examples, how impressive the intraretinal transduction is. Remember, this is just AAV2, but we're getting full thickness transduction of the ganglion cells and many other cell types, including Mueller glia, some photoreceptors in the outer retina. In this example, you can see the sub-ILM injection was performed adjacent to the fovea, but seeped into the fovea. So we're excited about this, and we've started to look at other capsid variants, and ideally we'd love to combine some of the variants that we identified in our library screen with this injection technique and see where we can get. But the beauty of sub-ILM injection, again, is that it overcomes the major barriers that we've discussed, the first of which is dilution in the vitreous. The second is potential neutralization by capsid antibodies in this non-immune privileged space. And the third is that physical barrier, which is the ILM. And we think this is a surgical approach worth pursuing because it can overcome all three of these hurdles. So I spend a little bit of my time outside the retina. Going to the front of the eye is like a different world, but I have done it, and the reason for this is because some of the leading causes of blindness are associated with defects in the anterior segment, including glaucoma. So what I'm showing you here is examples of primary open-angle glaucoma and closed-angle glaucoma. So you have a substance called aqueous humor that is replenished constantly and leaves your eye via a tissue called the trabecular meshwork. And normally that humor will leave, but as we age, sometimes changes in the matrix of that trabecular meshwork prevents that aqueous humor from leaving the eye. And even though this angle is open and that humor can flow freely, it's not leaving the eye. And as a result, your pressure in your eye goes way up. And this is a leading cause of blindness. It affects people, about 10% of people over the age of 80. And again, is characterized by, in most cases, elevated intraocular pressure. So in collaboration with a colleague at MSU, Andras Komarami, who has spent years of his career characterizing a beagle model of primary open-angle glaucoma, we decided to try and cure this model of glaucoma. So this dog model is very similar to the human primary open-angle glaucoma, and it results from a mutation in Adams TS10. This is a gene that encodes a protein heavily expressed in the trabecular meshwork, and we know it's important for the formation of extracellular matrix and microfiber structures. Let me go back one second. Just show you a feature of this dog is what we call optic nerve cupping. So as that intraocular pressure rises, what it does is it puts pressure on the ganglion cells at the back of the eye and the retina. And when that happens, you get a cupping of the optic nerve head. So this is a healthy optic nerve head. It sort of looks like a volcano. These are all the ganglion cell axons leaving the eye, going to the brain. In a glaucomatous eye, you can see that a lot of pressure has been put on that. And so it's literally cupping. So we set off on the pursuit to find AAV vectors that could efficiently target this tissue. And for a while, AAV was not used at all to target this tissue. It was thought that adenovirus and lentivirus were the only ones to get it done. And then for a while, it was, well, only self-complementary vectors can get it done. But we thought with all of these advances in designing new capsid variants that we could find one that could be used in a single-stranded form to deliver a gene to the TM. And we identified an AAV2-based vector containing a single YF mutation that got that done quite effectively in rodent. And what we found also was that our results in rodent were quite predictive of what we could achieve in dog. So this was really exciting, very new data, but what we found in several, I'm just showing you two examples, but several of these dogs was that when we injected vector at about 5 weeks post-injection, you start to see a drop in the intraocular pressure in the treated eye. And this effect has proven to be stable over the lifetime of the experiment, so we're past 72 weeks at this point. So this is the first time anyone has shown a correction of a phenotype in a model of glaucoma using a single-stranded AAV vector. And we also found up to a 92% increase in outflow facility in those treated eyes. And I got this data yesterday from my colleague, but this is just to show you that optic nerve cupping phenomenon that we talked about. This is an untreated eye, Adams TS10 dog eye, and you can see the optic nerve head is cupped. There's been ganglion cell loss in this eye clearly. But in the AAV treated eye, you can see that nice volcano structure that I referred to. So we are not only increasing outflow facility, but we are changing the dynamics of what's going on in the ganglion cell layer. And this is the ultimate target, right? We want to save those ganglion cells so that we can prevent vision loss. And again, as I mentioned, the therapy has been stable for over 72 weeks. So that's the end. I made it on time. But I have a lot of people to thank, everyone in my lab and their silly children, and also Sergey and Mavis and Arun Srivastava. I have the best colleagues at UF. I couldn't do any of it without them. My clinical colleagues at the University of Pennsylvania, Sam Jacobson and Art Sidisian, who are constantly characterizing the patients and telling me, Shannon, these guys are worth treating, go after this disease. My fantastic collaborators at the University of Alabama at Birmingham, especially Paul Gamlin, who's a primate neurobiologist and helps me a lot to characterize the transduction of these novel variants in non-human primate. And then, of course, Andras at MSU, who's been integral in the glaucoma studies. So with that, I'm happy to take any questions. Thank you.
While we're waiting for people to queue, I'll ask a first question. One issue with administration to the eye is pressure during administration. I'm curious if that can be helpful as well to help get things across the inner membrane.
So purposefully increasing the pressure to
Right.
get over that membrane?
I'm just wondering, so you're always going to have some amount of pressure when you're administering. And,
Right.
what's the sort of allowable pressure, and is it some amount of pressure okay?
Typically in monkeys, we'll inject about 200 microliters into the intravitreal space. Paul can correct me on that if I'm wrong, but that is typically allowable without any significant increase in pressure. And we've never done an experiment to evaluate whether purposefully increasing
Just
the
a little
pressure.
bit of pressure.
Yeah, no.
Okay. If there are no other questions, we'll move on.